Hey everyone, so our names are Andrea and Tulip, and today we will be teaching you about the polymerase chain reaction, also known as PCR. And to make it easier, we're going to compare it as if it was a recipe. So we're as with any other recipe, you need your ingredients. So what do we need to replicate DNA? And our procedure or method, which is how does PCR work and how do we get to the outcome, which is our amplified DNA. Lastly, we're going to be talking about the applications of PCR and how it relates to real world situations, and we're going to have a worksheet at the end in order for you to see if you clearly understand PCR. And so with any other recipe, you need to use the right ingredients to make sure that your outcome turns out the best way it can be. And so Tulip is going to talk about the ingredients in the next slide.
Hi guys, I'm gonna talk about the components needed in a test tube to start a PCR amplification experiment. So the components are DNA template, primers, DNA polymerase, and DNTPs. So for DNA template is one of the main ingredients for the PCR reaction. The new fragments made during the process of PCR also act as DNA templates. Production of DNA takes place when DNA polymerase enzymes attach to the template. The result of this process is that a large number of copies of specific DNA are made in less time. The template of DNA is the most purified DNA that is free of any kind of chemicals or DNA contaminants. If the composition of the DNA template is inappropriate, then it can lead to the failure of the reaction. It also plays a role in the functioning of DNA polymerase called DAQ, as it can also be activated if DNA template is recognized as a substrate for the PCR reaction. DNA template is a major requirement for amplification as it is not possible without it. The direction of the DNA template strand is from 3' to 5', and for the daughter strand, the direction is from 5' to 3'. As you can see on the next slide, we have a little bit animation for you where it describes the two categories of a DNA template, which is the target sequence DNA, that is the amount of the DNA to be amplified, and the non-target sequence of the DNA. So as you go on the next slide, you can see that DNA polymerase has some general characteristics, which are like speed, fidelity, specificity, and many more. DNA polymerase is an enzyme that plays a role in the synthesis of DNA. Before the division of cells' DNA into its two copies, it is copied by DNA polymerase. The role of DNA polymerase is to add nucleosides after it is attached to the end of the primers. DAQ polymerase is the main polymerase used in PCR. It is isolated from the heat-tolerant bacterium called Thermus equites. This bacterium can be found in hot springs. It can also has the ability to be stable in the heat conditions and can be observed to be most active around 70 degrees. The ability to stay heat stable helps in the PCR reaction as it is heated several times to denature the DNA template. For the next slide, we have a little bit process picture for DNA polymerase as it goes through all the processes in order to form a DNA amplification. And next thing is the primers. Starting point of the DNA synthesis is primers. Types of primers used in PCR reaction determine this quality. The supreme primer pair depends upon the information of the DNA sequence. Single strands of DNA or RNA sequences are called primers that have length of 20 to 30 bases. One main quality of primers is that it is heat resistant. When a PCR reaction is cooled down to 50 to 60 degrees, a primer is able to connect to a DNA template with the help of hydrogen bonding. The primers are a short sequence of nucleotides that are complementary to each other and the end of the DNA. The formation of complementary strands of DNA are made up of the polymerase enzyme only after primers are bound to loose DNA bases present at that time. And then, due to the presence of the DNA strands in the opposite directions, formation of forward and reverse primers takes place. The direction of the 3' end of the two primers is facing towards each other. To amplify DNA, a specific region is flanked by the primers. For the next slide, we have did a little bit animation for you guys where we have shown what is a DNA template and a DNA daughter strand and how does a primer binds to the DNA template, which is always in anti-parallel direction. So in the last slide, we're going to talk about the 4 DNTPs, which are DATP, DGTP, DCTP, and DTTP. So deoxyribonucleic triphosphates or nucleotide bases such as ATGC are required by DNA polymerase to generate DNA. The amount of each DNTP that should be included in a reaction should be about 20 to 200. If they are added in excess, the polymerase activity is inhibited, whereas if they are in less quantity, the fidelity is enhanced in polymerization, but the application yield is reduced. Increase in DNTP's concentration is required when they are no longer target sequences. And after the three DNTPs are linked together, it results in an unfavorable state. The energy is released when the outermost phosphate is removed. Monomers are linked to the polymers with the help of this energy. The reaction of PCR is carried out by the energy that is provided by these activated monomers. The DNTPs such as adenine, guanine are all called purine bases, whereas cytosine and thymine are known as pyrimidine bases. And we all know that free nucleotides are used in the process of building DNA sequence and for DNA polymerase. And in the next slide, as you can see in the animation, that we have tried to show you all the deoxyribonucleotide, triphosphate, diphosphate, and monophosphate. I hope you are clear about the ingredients, and we would now learn more about procedure of PCR.
Okay. So, now that we know more about PCR and the ingredients that it needs, we're going to be talking about the procedure and how it actually works. So we have 3 steps. 2 of those steps, step 1 and step 2, is going to be the preparation state. And think of it as if you were cooking and you're making a stew, and you need to cut up all your vegetables and your meat and whatever before you put it in your stew, before you put it in your pot. So same thing with PCR, you need to prepare your DNA template first before you can replicate it. So the first step is denaturation. So denaturation is when you're heating up your DNA to about 95 degrees Celsius. And we know that our DNA template is going to be double-stranded, and what's going to happen is it's going to separate these 2 strands apart from each other, and we need this to happen so that the primers can bind to the DNA, which I'll be talking about in the next step. So in the video you can see that the double strands are initially together, and after heating it up it's going to be separated, and this will happen this will take about 30 seconds to happen. So the next step is called annealing, and after you heat up your DNA, you're actually going to be cooling it down to around 50 to 70 degrees. And after we lower the temperature, the primers will be able to bind or anneal to the DNA, and the primers are going to make hydrogen bonds through base pairing with the DNA template. So how do the primers actually know where to bind to which part of the DNA? So we can see it in our video that our primers are binding to the DNA, but as Tulip mentioned, we know that our primers are going to have its own directionality as well. So it's going to have its 5' and 3' end, and we know that it has base sequences on it, and what's going to happen is the base sequence is complementary to the actual DNA template. And so you can see that the primer is binding to the DNA template and it's complementary, and the other primer is also binding to the other DNA and it's complementary to each other. And you can see that the 3' ends are going to be facing each other. So now that we're done preparing our DNA, we can go to step 3, which is extension. And this is where the actual replication happens. So the DNA polymerase binds to the primer, and it's going to add DNTPs to make the new strand of DNA. And it adds free nucleotides from 5' to 5', or in other words, it goes down the DNA from 3' to 5', and this results to the copy of our DNA. And you can see this in our animation where our primer is bounded to the DNA, and a DNA polymerase, which is not part of the video, is gonna just go down the DNA in order to make the base pairing of the template DNA. And this process is repeated again and again to amplify a certain sequence in our gene. And if you do this for about 2 hours, you get about 1 billion copies of the amplified DNA. So, in order to understand PCR better, we can try applying it to real life situations. And a really relevant example that PCR is used in is during COVID, and so Tulip is going to talk more about that.
Hi guys, so I'm going to talk about COVID-19 and PCR tests, but before that I'll talk about COVID-19. So it's an infectious disease caused by a virus. Most people who experience the disease recover after mild symptoms. When a person sneezes or coughs, the droplets are transferred from one person to another. The weight of droplets is heavier than simple droplets, which cause them to fall on surfaces quickly. Cause of COVID-19 is that infection COVID-19 is caused by the virus strain of severe acute respiratory syndrome coronavirus 2, which in short is called SARS COVID-19 2. The virus that caused COVID-19 belongs to the family of viruses called coronavirus. Researchers have found that antibiotics don't work against viruses. People suffering from COVID-19 can form bacterial infections as well. So some signs and symptoms for COVID-19 are like fever, dry cough, tiredness, and difficulty breathing or shortness of breath, chest pain. And so we tried to make a connection between PCR and then COVID-19, because as we know, world is facing a pandemic and it is important to know how to prevent it. The first step is to prevent it from spreading, as it is a contagious disease. By preventing the interaction of infected people with healthy people is by doing a COVID test. The nose swab PCR test is used for COVID testing and is one of the reliable tests. So if results turn out to be positive, it means the person has the disease. What is PCR test? So it is a test that is used to test genetic material which is derived from particular organisms such as viruses. The test helps one to know if there is presence of virus in the body. The test is reliable as it can be even detected by the fragments of virus present in the body after the host has healed from the disease. So what is a COVID-19 PCR test? COVID-19 PCR test is used to analyze the upper respiratory specimen, which is performed in order to find the genetic material of COVID virus in the body. COVID-19 PCR test is also a molecular test which is used to amplify the small fragments of RNA which are present in the DNA. If SARS is present, it is detected during the amplification of RNA that is present in specimens. So what are the key steps for PCR? To find the result, a PCR machine called thermal cycler is used along with some chemicals and enzymes. The target genetic material present in the test tube is increased after each cycle of heating and cooling. Millions of copies of genetic material of viruses are formed in the test tube. Chemicals that are present in the test tube turn the sample to fluorescent color if a presence of virus is detected. After the amplification of the sample, the signal is detected by the machine. After the detection, scientists can determine whether the result is positive or negative. So this was our real life application, and now Andrea is going to talk about the limitations of PCR.
Although PCR is very helpful, it still has some limitations that it can be worked on. And so this is mostly in the clinical setting, as we mentioned that it is used to diagnose infectious diseases, and one of the things that can cause stress to a family is getting a false positive. And so this is when the virus or the disease is detected even though it really isn't there. And the reason why this happens is because previous DNA from a previous sample can be carried over, and PCR can be quite sensitive, and so that's going to amplify that part of the DNA as well, causing to result into a false positive. Another example is that we know that we need to know the specific primers needed in order to replicate the DNA. And so doctors or physicians and clinicians and researchers need to know which primer they need to use in order to start that replication. And lastly, this can be more expensive compared to other methods of diagnosing. So that's it for our presentation. Thank you so much for listening. There is a worksheet that goes along with this video, and you should be able to answer it based on the explanation from this video. So yeah, thank you.